When we think about medicine right now, most places in the world, we practice reactive medicine. We wait until you get sick, and then we go and treat it. It's incredibly inefficient to do it that way. This is a beginning, along with genomics, where we can start being, I think, proactive. Let's go find the people who are going to be sick, and let's treat them before that disease ever happens.
Hello, and welcome everyone. I'm Jesse McQuarters, editor of StatBrand Studio, and this is a special collaboration between SBS and the Nucleus podcast from Regeneron, a show which explores how biology and technology converge to shape the future of human health. Today, we're diving into one of the most dynamic frontiers in human biology, proteomics, the large-scale study of proteins and how they interact within our cells, tissues, and systems. To help us unpack this, I'm joined by Dr. John Overton, Vice President and Chief Sequencing Officer at Regeneron Genetics Center. We'll explore how advances in proteomics and multi-omics are revealing new biological connections and shaping the future of personalized medicine. John, fantastic to have you here.
It's great to be here. Thank you, Jesse.
Yeah. By the way, you have one of the coolest titles, Chief Sequencing Officer. Sounds like amazing. Tell me a little bit about what RGC is. Tell me what you guys do.
Yeah, so the RGC was started about 12 years ago with this grand vision of we were going to look at people's DNA and we're going to find the individual differences between two people that either made them susceptible to or resistant to developing disease. This was going to give us a better understanding of biology, give us a better understanding of diseases, and hopefully eventually lead to new therapeutic targets to help people. It started out as a very small operation, and over the last decade plus, we've sequenced over 3 million people, which is just a really monumental resource.
And give me a sense of the complexity involved, because I know, sure, there's maybe four bases, right? It's like at its most basic level, it's not that complicated, but you layer in all these people and all these different combinations. I'm sure it's enormously complex.
Yeah, it's A, T, Gs, and Cs. So fundamentally, sequencing the 4 bases of DNA, we saw this long trajectory where we're just going to have to make really, really big databases. And we wanted to stay ahead of that. But in terms of complexities, it's a lot more than just A, T, Gs, and Cs, because we want to sample that full diversity of people around the world. And so a lot of sequencing has been done from very focused areas in the world, but the RGC, we want to sequence everywhere. So we increase the complexities by having over 150 different collaborations around the planet where we're getting samples from.
Those samples I know you mentioned, they were from around the world. Are there certain biobanks? Like, where are you deriving all this data from that's informing this?
Diversity is incredibly important. The largest project that we've done to date is a project called the UK Biobank. It's an incredible project. And so this was started about 20 years ago, and it's 500,000 volunteers in the UK. They signed up for the study. They gave a DNA sample. They gave their blood. They subject themselves to multiple different forms of imaging, all knowing that this would be made publicly available to scientific researchers. And so we did a certain type of sequencing on that several years ago. And that's now been probably one of the most widely used resources in the world. And now we've just started doing the proteomics on that data set as well. It's the same original 500,000 people, and 100,000 of them came back. And so we've got this longitudinal data where we see their baseline, and now what does it look like 10, 15, 20 years later? And they're doing imaging. So if you think in diseases like Alzheimer's disease, we've got an image of your brain from when you were 40, and now we have an image of your brain when you're 60, along with your proteomic changes. So we can start integrating all of this data. And the really incredible thing about the UK Biobank, it's been paid for the data processing by a group of pharmaceutical companies, but it's made publicly available to the world.
So we're going to talk about all the omics, at least a little bit. Tell me a little bit about proteomics first. What's its value? How does it differ from metabolomics and some of the other kind of levels of looking at human data?
Proteomics is incredibly interesting. It's a relatively new field. It's only been around for a few years now, and really came along behind the sequencing world. So as in advancements in next generation sequencing, as those costs got lower and lower and the throughput got higher, it enabled new assays like proteomics. Proteomics, it's the natural progression. It's the next step. If we think of DNA as the instructions, those are the instructions for how to make the proteins. And so as we're amassing these huge databases of DNA, we're finding these individual changes. And we're trying to predict, what does that change do to a protein? For many, many years, it was a guess. We'd look at other parts of the literature, make guesses on the changes that happen, but now we can do proteomics. And so we see these individual changes in the DNA, and we have this new assay that we can measure proteomics. And so now we have these real world outcomes where we can measure what's going on from that individual change in your DNA.
And give me a sense of scale, like how many base pairs go into defining, kind of a basic level protein?
Yeah, so in proteins right now, each individual protein is measured through one of these assays. We tag it with DNA as we're measuring it from serum or plasma or various other sources. It gets tagged with the DNA barcode. We put that on the sequencer and we count it. Right now, the assays we're using, they'll look at several thousand individual proteins that are in your blood, and we see those as a sequencing output.
Certainly, we're layering in protein data. We're layering in, things that influence metabolism, imaging data. Tell me about how all of those pieces layer together and kind of give you a more complete picture of human health.
Yeah, it's really critical to have as many different data resources as possible because we don't want to miss anything. And we start with the DNA, we sequence that, and we get the fundamental changes between two people. Maybe the proteomics give us some measurements. The imaging is incredibly useful now because we can start seeing changes in people many, many years before the disease ever manifests itself. So we have these multiple assays where we can start looking at disease, whether it's the natural progression of how that occurs or preemptively seeing it and predicting that you get those diseases.
So tell me what kind of picture emerges? What have been some of the success stories? What are some of the things that you're seeing now that you're layering all these different parts together?
We found some incredible things with the proteomics right now is that where you think about a disease like cardiovascular disease. Cardiovascular disease, gold standard, LDL cholesterol. Everybody knows. You go to the doctor, you measure LDL cholesterol. But there's still people who have perfectly normal LDL cholesterols, they still get cardiovascular disease. We look at the proteomics or the genetics, we can predict that maybe this person with perfect LDL cholesterol, they're also at a predisposition of developing cardiovascular disease later in life. And so we can start looking at these things, build up our case numbers more because we're using different types of methods to characterize either the early onset or the progression of diseases.
And what's the balance of kind of human intervention and just raw calculations and sort of computing power that you're just, throwing at this to try and, tease out some of this probably very, very intricate data?
We've solved the sample processing side through robotics and automation. And as we continue creating these huge data sets, we're learning more and more how to create massive ways of looking at this data. And we're constantly having to develop new computational approaches to do that.
Obviously, again, huge amounts of computing power. Is that a processing center on site? Is it cloud computing?
When we started the RGC over a decade ago, the way most places did this was on-site compute. You built huge banks of computers and you did your analysis. We just knew that wasn't going to be enough. It was going to have to be bigger. So we were probably the first genome center that went fully to the cloud. We've never had on-site infrastructure. Every time we sequence a sample, it gets pumped currently into AWS, the Amazon Web Services. We do all of our compute there. It's got this elasticity, so we can spin up so many computers at a time to be able to analyze data sets like this.
All right, and kind of a geeky question, but like how much data is stored on those AWS servers?
Many, many petabytes, huge numbers of samples.
And tell me a little bit more about the logistics involved. Is there different machinery used? I know we talked through it a little bit, but when it comes to sequencing proteins, does it take more time? And how many proteins are needed each time?
So the proteomics is a very different assay than we're used to in the DNA world. In the DNA world, we take your DNA, we chop it up into little pieces, we put it on a sequencer, and we read out the ATG it sees. For the proteomics, there are multiple different ways to do this, but essentially you have some sort of thing, an antibody, an aptamer, which is a group of amino acids that binds to a protein and it has a tag on it. When it binds to that protein, we're able to amplify that tag, and then we throw it on a sequencer and we just count the number that are there. The number of tags is the number of the proteins. We're in an incredibly interesting era now where new approaches to proteomics are coming out, but also the sequencing world is evolving. And we're able to do these assays on different platforms and not only drive the innovation, but help with cost too. And so with the proteomics, I think more so than in the past, we've had these tools at our disposal to really push that technology forward.
Yeah, one of the things that always strikes me about Regeneron, so interesting, is obviously it's a commercial entity, but it really seems like the scientists and kind of the thinkers of the organization have some pretty wide latitude to explore these things and then also think ahead. What capabilities do we need that maybe there's not a direct application for now, but have it be a capability in the future? Yeah. Tell me about that.
Yeah, for sure. I mean, innovation drives the company, and so does science. We're always focused on the science and how can we do it better. and that I was incredibly fortunate when I joined the company. What I was allowed to do is had an empty building and was told to go build something. Tremendous offer, but I was able to take those risks because the senior management is so focused on innovation. They want us to try something new. And doing something like fully automating or going to the cloud, those are big risks. Nobody did before, but that's really the culture at Regeneron.
All right, excellent. So moving kind of into some of the practical insights and applications, from a research perspective, what are some of the challenges in integrating proteomic data with genomic data, especially when you're looking at preclinical and clinical studies? Mm-hmm. And for example, are additional samples required for proteomics research, or can the same samples being collected for genomics research be used for both?
Yeah, well, I'm envisioning as we're moving forward that every person that we sequence, we do a proteomic sample on them as well. The costs are coming down, the throughput is going up, where this is certainly becoming a reality. In terms of challenges, we got to control for things like when you go into the office to get a blood draw. In proteomics, I can tell if you had a donut on the way in. We have to be careful about comparing those types of samples and seeing what's happening. We need to be careful about controlling as many things that we can, just to make sure that when we're making these comparisons, they're very fair when we're looking at them. But it's also thinking about the question. And so if we give you, say, a weight loss drug, we'll take a sample before you start. How often should we sample in the future to see what happens? And these are incredibly interesting questions because you can look down on the scale and see you lost weight. But what did it do to your health? Are you actually healthier? Then we're thinking things, and we've seen data now that the proteomics can show changes that you don't see necessarily by standing on a scale.
I know you were one of the founding members of RGC and really integral into its growth over the years. Could you have imagined what it's grown into when you first started RGC?
I mean, our original goal is we thought we were going to sequence 20,000 people a year, do that for 5 years, and have this incredible database. We got through 20,000 samples in the first 6 months. 100,000, in the first couple of years, we knew this thing was going to have to be bigger. I didn't envision it could be as big as it is today, where we have incredible ambitions. We've sequenced 3 million people, but we want to go to 10, 20, 50 million people because we really don't know what we're looking for yet until we find it. And so it might be that 50 millionth person that we sequence where we find that really X marks the spot moment where we're able to cure a disease that had no treatment before.
In 5 years, you might look back to something you collected 10 years ago and be like, oh, it was sitting there all the time. We just had to look at it in the right way in a certain light. And
5 years ago, I probably wouldn't have predicted you today that we'd be doing the world's largest proteomics project. I remember sitting there with the vendor we've partnered with on this. We're sitting on my patio because 5 years ago, we're in the midst of a pandemic. And so we're trying to figure out what can we do now to set this up to be ready for the future. And it was a perfect example of the risks we take at Regeneron. At risk, we built automation that we thought would be ready for when the world was set for these proteomics projects. We were fortunate that it all worked out, but once the technology was available, we were right there ready to go.
We kind of talked about treatment up to this point. Tell me a little bit about how it might prevent disease and again, kind of identify who is at risk for something.
I think proteomics is the wave of the future for this. Because if we think about medicine right now, most places in the world, we practice reactive medicine. We wait until you get sick, and then we go and treat it. It's incredibly inefficient to do it that way. This is a beginning, along with genomics, where we can start being, I think, proactive. Let's go find the people who are going to be sick, and let's treat them before that disease ever happens. Because when we think about the proteomics, we can take your blood sample. We can take it once a year, take it as part of your physical, and we can probably start detecting diseases ahead of time. If you think about diseases that take a long time to notice, something like liver disease. Your liver is a really strong organ. You can have liver inflammation for a long time before it goes detected. There have to be proteomic signatures early where we could see this and start treating you. Think about other fields like women's health. Something like endometriosis. Endometriosis can go a very long time without being treated. There's a lot of pain, there's a lot of discomfort before there's an invasive approach to go see what that disease is. There have to be other signatures we can see ahead of time. And so we're very optimistic that by understanding people's proteomes, we can sense diseases earlier and then start those treatments before they progress into something worse.
And I think cardiovascular risk is huge in the U.S. I mean, it affects so many people. Some are genetically driven, but some are lifestyle driven, and maybe you can start to understand some of those risk factors and help people live longer and be healthier.
Oh, for sure. We've already seen from the proteomics, we put together risk scores. So this isn't looking at individual proteins. This is looking at groups of proteins and how they move with certain diseases. We can do this with something as simple as age. We can predict your age very accurately by your proteome. And we can look at things like your health. And we can say, you are 44, you have the health score of a normal 44-year-old. What if you are 44, but you have the health of a 60-year-old? That's when we go say, okay, there's probably something else going on. Is this person unhealthy? And we've started doing things like this, and people who fall off that line, they have cancers. They have other very high-risk diseases where we can see that they do have underlying health issues that can be predicted by proteomics.
Is there a point where there's too much information? Where okay, like you're really thinking all these different things that could happen. I'm at risk for this. Obviously, only a certain proportion of people will that will be realized in those people. How do you balance those sorts of factors?
People want to know more and more about their health. And I think genetics and proteomics will be one of those next waves where they're going to see this information and they're going to want to know, what can I do? Is there something else I can try? Or how can I help my family members?
I know you've got a number of collaborations with research centers and industry and technology partners. Which are the most critical to advancing the field?
So in terms of proteomics, we've done a very large-scale collaboration with our great partners in Geisinger. Geisinger is a healthcare system in Pennsylvania between Pittsburgh and Philadelphia, covers a few million people. We're doing proteomics there. We've already sequenced them. I think that'll be an incredibly interesting project because we can bring together the DNA sequence variations, the proteomics, and start helping people in that sense. We sequence everything at Regeneron for research, but Geisinger then validates that data in a clinical setting, and they get it back in the patient's hands. I think when we start layering the proteomics on that, it will make it even more powerful as they figure out the best way to treat their patients.
And are they looping back to you and kind of influencing how you collect data and store it and communicate with them?
Yeah, and Geisinger actually, they put all of this on their website. It's a project called MyCode. They're continuously updating their statistics on how many samples have been sequenced and how many data points that they've returned back to their patients so the world can see the impact of how this is going.
What barriers need to be addressed in terms of technology, data sharing, collaboration, to really fully unlock the potential of combining protein, genetic, metabolic, and imaging data and whatever comes in the next 10, 15, 30, 100 years?
Yeah, a lot of it's going to be access to the data. And that, again, back to the UK Biobank, this will be made publicly available, and that it's being funded by pharmas. But it won't be until it gets really into academics' hands where they'll have huge access to it, and they'll make discoveries that we haven't thought of, and they'll come up with innovative new ways to analyze the data, bring data sets together. That's going to be a huge next step to fully appreciating the value of this.
That's where I feel like having a company that's run by scientists, the driving force of scientists, that speaks to that human impact that's happening down the road.
Yeah, and it helps too on the technology side because we have early access to these things. We can start thinking about them, and we can play with these workflows to make them more simple so that we can get that out to the masses. The way we've built this at Regeneron, it's a very large scale. It's unrealistic to think everybody in the world is going to be able to do this. But how do we get this in every lab? How do we get this in the hospital setting so people can start generating this as part of routine clinical care? And it will take some of us taking those very large scale assays, miniaturizing them down, making them more simple so that everybody can run them.
So I know you talked about some of the collaborations and partnerships that Regeneron is undergoing, really internationally. Can you tell me about some of those and how you're really powering science throughout the entire world?
We have one incredibly interesting project we're going with right now. It's a collaboration with a group called H3 Africa and with the University of Michigan. In there, we are sequencing samples and doing proteomics on samples from Africa. But this project also has an incredible additional component. We're bringing scientists to Regeneron. We are training them. They're learning how to do the methods that we have so they can go back to Africa and they can perform these types of assays there and they can communicate the data back and really hopefully make a huge impact on site.
So looking forward in the next 5 to 10 years, I know you mentioned, really delivering, data and this picture of health into the hands of clinicians and patients. What else are you excited about? What else are you looking forward to?
Yeah, I really think the proteomics is going to be the wave of the future. And what I really want to see is this collected as part of routine healthcare because we want to see it over time. What happened before you developed disease? What happened when you got it? And what happens when we put you on a treatment? We can think about these as not only as predicting human health, but as biomarkers for various diseases. So people don't have to undergo expensive imaging, uncomfortable biopsies. Diagnosing liver disease, it's a pretty big gauge needle to get some cells out and look at the inflammation. What if you don't have to do that anymore? How many times have you yourself said, I don't feel like going to the doctor. I don't want to go do these tests. What if it's just a blood test? What if I can send you a kit in the mail? Just do it at your house. send it back in, we measure it, and we can get back to you. I think it's going to just have an incredible impact on how we perform healthcare.
All right. Well, John, this has been a fascinating, enlightening conversation. So appreciative of you taking the time to share your work and the work of Regeneron Genetics Center and Regeneron writ large. I know STEM and education is a huge part of what Regeneron is all about. How can young people that might be hearing this kind of get involved with RGC and start to dive in some of this fascinating world of genetics and proteomics?
Yeah, no, and Regeneron has a huge commitment to STEM education and just getting the sciences out there in general. My biggest advice to somebody thinking about this field is when I came through college, you either were a biologist or you're a computer scientist. Now those fields have melded and we have bioinformaticians where they understand the biology, they understand the statistics, they know how to code, they can write computer programs, they know how to take advantage of AI. Keep doing that because these data sets are going to get bigger and bigger and bigger where I can't look at it anymore and just use my brain to find the insights, is that we're going to need them to develop new technologies and new algorithms to really expand our understanding of how this is happening.
Yeah, fantastic. And for our listeners and viewers, we invite you to follow the Nucleus podcast on your favorite platform, share it with your friends and colleagues, and to visit regeneron.com to hear more about the organization's work at the forefront of medicine. I'm Jesse McQuarters, editor of Stap Brand Studio, and thank you for joining the conversation today.